Hello everyone, I'm Matt Yoder. On behalf of my co-authors and myself, I'd like to thank you all for this opportunity to present at Digital Data 2023. I want to start by acknowledging that, of course, the things you hear about and the tools that you hear about today are not just mine. They're the product of a lot of different people, in particular the Species File Group. We're a small endowed collective that's building out biodiversity informatics tools. I also want to note that really exploring the ideas that were first published in the first edition of the Bulletin of the Society of Systematic Biologists. So go check out that edition of the Bulletin if you want to read more. Throughout the talk, I'll be referencing TaxonWorks. This is a web-based workbench for serving the needs of sort of biodiversity informatics science. I'd encourage you to join our Wednesday meetings. We meet every Wednesday unconference style. It's an open, whether or not you use TaxonWorks, and you come learn more and ask questions or see it in action if you wish. That's every Wednesday. And you'd also might want to join us in late September or October of 2023 for our fourth, I think, TaxonWorks Together, a sort of conference on all things TaxonWorks and related topics. So today I want to think about how monographs can sort of inform us or sort of tease out some ideas about how we work with digital data in general. For those of you who are not familiar with a monograph, it's sort of a comprehensive treatment. It's all we ever know, or it's sort of an effort to produce all we ever know about a taxa and put it in a single place. And really we're looking at the process of producing those monographs. So how do we go about creating monographs, and then how is that process similar to how we work with digital data in general? So throughout, I'm going to reference just a couple of steps or things we do in monography, and then I'll provide a couple of examples of how those steps relate to things in TaxonWorks. So to start off with here, we see that when we start building a monograph, we're really thinking about integrating data from a lot of different sources. And in TaxonWorks, we have a very rich data model that serves the needs of monography in general. And that data model doesn't, of course, catch everything. And in TaxonWorks, we have ways to extend that data model with controlled vocabulary terms in a lot of different ways. And you can think about each monograph as treating a different group of taxa, a different group of diverse data, and they're going to necessarily need fields or data types that aren't necessarily universal to all taxa. And the extensible vocabularies that we have in TaxonWorks let us kind of capture out those rich data. Throughout the monographic process, we gather data, collection objects, for example, from collections. We gather sources, we gather observations. One of the ways we can do that in TaxonWorks is through the Darwin Core importer that takes the rows of Darwin Core data and it sort of translates them into what TaxonWorks models data as sort of things. So not necessarily rows or fields, but actual instances of classes of data. Here we see instances of people that are linked into a collection object. And these things as a foundation really let us do some very rich things as we go through the process of building out our monograph. Monography is longitudinal. By that I mean that it is a process that treats everything that has happened for a taxon over time. So we have to cite all of the nomenclature, all of the specimens, etc., that are known for that taxa, whether or not they're 250 years old or 100 years old or were collected yesterday. And TaxonWorks lets us cite everything, all different types of things, specimens, names, collecting events, you name it. We can link it to a source and attach it to a citation. We'd like to point out that people are things in our graph as well, and citations let you link sources to people. It's one of the ways that we can link our data to people, and this lets us sort of give credit to people where credit is due in a kind of very rich way. When we get all our data and we've gathered all our data together, we want to relate them, and that's really what provides a rich context that defines our taxon. Here we see that we can create biological associations between specimens, for example, or between taxa and specimens. And so we can also create relationships between nomenclature. The nomenclature in TaxonWorks is stored as a big graph. You're seeing over 1 million assertions about the nuances of the ICZN nomenclature here on leafhoppers here on the right. And that rich relationship then creates the context for our taxa. As we're building out a monograph, we're observing, we're gathering in new facts, we're making new measurements, that kind of thing. We have the ability to create matrices in TaxonWorks of data by scoring taxa or collection objects. We can also score things like extracts. And one of the things we can do is relate those data to images through what we call a depiction. So images can be used to relate to not just collection objects, but anything in the system. As we build out a richer variety of data, we want to start thinking about how are we going to diagnose those taxa so that we can present that out in the monograph. And TaxonWorks has things like interactive key engines where we can sort of test our concepts by running them through the key so that we can keep refining them and fleshing them out. We also commonly think about things like things such as distributions. Does this map of specimens and sort of asserted distributions of historical taxa or distributions of taxa in the past, does this represent one or more taxa, for example? And we sort of are balancing these and looking at that, and TaxonWorks' rich GIS capabilities can let us do that as well. And we don't just do this kind of thing linearly. We do this over and over and over. We observe, then we refine, etc. We relate, and we keep doing it back and forth. We have things like unified filters in TaxonWorks that let us kind of query down in a myriad of different ways to get to the sort of core facts to see if they really support our hypothesis. We also have hundreds, there's well over 400 different ways of validating our data, something we call soft validations. And these you can think of as guides towards creating consistency across our data. Maybe you're missing this thing, or maybe the two things that you've asserted in the system don't logically make sense. And so you constantly kind of see this report that can help you make your overall product more consistent and of a higher quality. Once you're done and you're ready, you've done all this whole process, you want to publish it, render it out. We can export to GBIF or Catalog of Life. You're familiar with those kind of processes. And we also have a new product, companion product, we're calling it, to TaxonWorks, called Taxon Pages, that does what it's called. It produces Taxon Pages. You can deploy these over things like GitHub pages. They feed off JSON APIs. The project is not specific to TaxonWorks. The species file group will be producing well over 40,000 of these individual taxon page treatments in sort of huge monographic treatments coming up later on this year as we move some of our old data into new systems. So to step back, we want to point out that monographs don't just happen. This process of integrating, gathering, citing, relating, observing, diagnosis, we don't just do that once, we do this over time. And in the digital data era, we need to think about how those digital data relate back and forth to one another as the process repeats over and over. In TaxonWorks, we have the ability to throw identifiers on all of our things, and those identifiers can say that, can assert that the concept that we talked about in Monograph 3, for example, is the same as the concept in the knowledge base over here. As we expose data, we also get lots of feedback coming back to our users and our scientists. And through the annotation system we have in TaxonWorks, we can record that as notes. We can create custom tags or custom attributes that allow us to persist sort of the outcome of producing that monograph and having that monograph looked at by other people. So to wrap up, there's definitely issues that the species file group are interested in pursuing. I don't really have time to get into them, but we're exploring adding new types of things that relate to anatomy and things that help you track the DNA component of sort of taxonomic circumscriptions from the voucher to the actual product that you're going to produce. We know that there's lots of cool technologies coming along like ChatGPT and digitization pipelines that we're interested in thinking about how we can work with. And of course, people are the key behind all of this, and we really need to advance a lot of social capacity as well in this process. So if you're interested in any of the things that you see up here, again, lots of opportunities to join with us on those Wednesday meetings. So in conclusion, I hope we've pointed out that a lot of the processes that go behind monography are not really special or unique to monography. They're processes that we need in any sort of digital data science or endeavor. And really what we've tried to do with TaxonWorks is to take those kind of processes and localize them into a single place where we can kind of experiment with them and help them evolve. So with that, I'd like to thank you for your time, and hopefully I have a little time for questions. Thank you.